Ladies and gentlemen, please give out your applause to welcome Dr. Doudna, Principal Yang, and the CEO of TangPrize Foundation. Good afternoon, ladies and gentlemen. It's great to see you all. Thanks for coming here. Today, on behalf of TFG, we would like to welcome every single one of you to be here. Known as Oriental Nobel Prize, TangPrize combines the innovation and connotation of art with science and technology. Not only is it the pursuit of cultural elements, but that of the value of progress and change. Today, we have this greatest honor to receive our guest from the 2016 Tang Prize winner, Dr. Jennifer A. Doudna, to present here for the exchange of information and ideas. I'm sure your curiosity will be well satisfied through Professor's profound experience and knowledge. Now, let's enjoy a short video about our guest.
For a long time now, scientists have been working on research to understand the genetic causes for a lot of diseases. Every cell in our body contains a copy of our genome with over 20,000 genes and 3 billion letters of DNA. Emmanuel Charpentier, Jennifer Doudna, and Feng Zhang developed a CRISPR-Cas9 system that can either delete or replace unwanted genes.
With this interest, we came to the CRISPR-Cas system. We identified bioinformatically 2 RNAs that were expressed in S biogenes. So one is a tracer RNA, and the other one is a CRISPR RNA of the CRISPR-Cas9 system.
To look for a particular sequence of letters in the DNA code of a cell. And when the protein finds that sequence of letters,
making a cut to allow us to then replace it with a piece of DNA that's no longer able to cause the disease.
The editing, in principle, could be done outside the body by removing stem cells of the blood from a patient, correcting them outside the body.
Then we can transplant the cells back into the patient, and the cells will be able to produce new red blood cells that would cure sickle cell disease.
Unlike previous methods, it can be used to target many mutated genes at once in cancer or other diseases which involve multiple mutations.
Improve plants, we can use it to screen for new traits that confer either viral resistance or drought resistance, make crops more robust, and be cultivatable even in very harsh conditions.
The opportunities with human embryo editing are enticing in a way, because one could imagine correcting a genetic disorder that would then never be transmitted to future generations.
Programmable in an easier fashion, and it's also less costly, and it's also more versatile. This is important to say, in the sense that CRISPR-Cas9 allows not only to act directly on the DNA, but also to change the expression of the DNA.
Agriculture, drug development, fighting disease, preventing congenital defects, and even curing cancer. A simple yet incredibly powerful new genetic technology, CRISPR-Cas9 system is revolutionizing today, and tomorrow is bigger than you think.
Today, we are lucky to have our Principal Yang, and the CEO of TangPrize Foundation to join us today. Now, welcome Principal Yang to give us some words.
谢谢大家，今天那么多学校的同学们，大家一起来啊 ！Dr. Jennifer Doudna, Mr. 陈振川执行长 of the TangPrize Foundation, and all the teachers and students welcome. We are delighted to have you here to participate in this event hosted by Taipei First Girls Senior High School. Thank you for coming. The Tang Prize was established in Taiwan to honor the individuals around the world who have made great contributions for all humanity. And today, we are fortunate enough to have the chance to invite a Tang Prize winner, Dr. Doudna, to our school to deliver a speech for us. Dr. Doudna is a prominent RNA structural biologist. Her invention is an amazing technology for editing genomes. And this technology allows human beings to make changes to their DNA and, more importantly, to cure genetic disease. I think this invention is just incredible. And I believe that such an invention must be a result of constant efforts, admirable beliefs, and humanitarian concerns. So I also believe that our students, as well as those from Jianguo High School, from affiliated senior high school of National Taiwan Normal University, Shida Fuzhong, from Chenggong High School, Chenggong Zhongxue, and from Zhongshan Girls High School, Zhongshan Nugao, will benefit greatly by Dr. Doudna's speech. And after the speech, there will be a discussion period when you are encouraged to ask Dr. Doudna questions directly. This is such a chance of a lifetime to talk to her in person. I hope you can learn a lot from her and get inspired, and that your views can therefore broaden. Thank you. 谢谢大家，欢迎大家今天来到现场，谢谢。
Thank you so much. Do we also have this honor to welcome the CEO of TangPrize Foundation, Mr. Chen, to give us some words.
大家午安，大家好。Professor Doudna, Principal Yang, teachers, most important young students. Welcome to the TangPrize Youth Symposium. Today we are very honored to welcome Professor Doudna here to engage in a dialogue with the young students. The TangPrize is an international award founded in 2012 by Samuel Yin, the chairman of Luntex Group in Taiwan. The prize is awarded in 4 categories: sustainable development, biopharmaceutical science, sinology, and the rule of law. All of the young students here today, you are a turning point in your lives. The choice you make today and tomorrow will decide your future. Professor Doudna and the other laureates were also young, just like you. You can choose to be inspired by their growth and their spirit, their experience and their ideas, and those particular qualities that made them who they are today. I myself look forward to a day when one of you may be standing on the stage at the TangPrize awarding ceremony, receiving an award for the honor of your country and of your school. Curious ever, since her childhood, on the remote areas of Hawaii Island, Hawaii State, Professor Doudna is now one of the pacebreakers in the field of biology. She is currently professor of chemistry and of molecular and cell biology at the UC Berkeley, and a Harvard Hughes Medical Institute investigator. She is also a member of the National Academy of Science, USA. She is now in Taipei, Taiwan, to receive the TangPrize for her work in helping to develop CRISPR-Cas9 gene editing system, which is truly a revolutionary development that will be looked back upon as a turning point in biology and human history. On behalf of all of us at the TangPrize Foundations, I would like to sincerely thank Professor Doudna for joining us today and sharing his life with us. Our thanks also go out to Taipei Girls Force High School for arranging such a wonderful talk. And finally, thank you all for attending here this symposium today. Thank you very much, and wish you all the best. Thank you.
Mr. Chen, please stay at the stage. We will have the gift exchange with Principal Yang. Today, the Tang Prize Foundation have also the gift for Taipei First Girls High School. Please remain your step. Let's welcome Dr. Doudna to have the gift exchange with us. Let's have a group photo together. Thank you so much. Let's welcome 2016 Tang Prize winner, Dr. Doudna, to have her story with us.
Hello, everyone. Thank you very much for that warm welcome. And I certainly want to acknowledge Principal Yan and Dr. Chern, and especially all of the students that are here today. You can't imagine what an incredible honor this is for me to have an opportunity to talk to you about a little bit about our science and about how I got started on my career, and how I would love to see all of you take that same path if you so desire. So what I thought I would do is to tell you a little bit about the work that we're doing in my laboratory at the University of California, Berkeley, and work that has led to this great honor of the Tang Prize, and especially to start by saying that I want to acknowledge my colleagues and my students that I've worked with over the years that allowed this really exciting science to happen. In my experience, science is a collaboration. It's not done in a vacuum, it's done in a community of people. And so I invite you all to think about that as you learn about science and think about what you might want to do yourself in the future. So for me, this story really begins with DNA. So DNA is the chemical inside of cells that encodes all of the information necessary for life. I learned about DNA when I was in 6th grade. I was about 12 years old, and my father gave me a book that was written by James Watson, one of the 2 scientists that was involved with other people in discovering the structure of DNA, which is a double helix. And ever since then, that was a discovery made back in the 1950s. Scientists all over the world have been thinking about this DNA structure and imagining that it would be amazing to be able to change the structure of DNA, or change the letters in the DNA code, so that we could change the properties of cells, or even do something like correct a DNA mutation that causes disease. So this is, again, just a close-up of the structure of DNA, and it's a double helix, meaning that it has 2 strands that wind around each other, and it's made up of chemical letters that come together in pairs. And that's very important because it ensures that the genetic information in a cell can be passed on to the next generation. So it's how life is really propagated, how life can go forward. So imagine that you could actually have a tool, a technology, that allowed you to go inside of a cell and make a very precise change to that DNA code. Basically, what if a cell's DNA could be edited just like the text of a document? If you saw a mutation in the letters of the DNA in a cell, what if you had a way to change those letters and correct them so they no longer caused disease? It would be an amazing thing. And our own research was initially not actually focused on this particular question at all. We were working on something seemingly very different, which was understanding how bacteria fight viral infection. This was a work that started in my lab at UC Berkeley about 10 years ago and led to a collaboration with Emmanuelle Charpentier, who is also being honored this year by the Tang Prize. And so I want to tell you very briefly about how a revolutionary genome engineering technology, meaning the ability to change the letters in the DNA code, how that technology came about with a project that was really about our own curiosity about how bacteria fight the flu. So it turns out that bacterial cells, like us, we can get infected by viruses. Well, bacteria can get infected by viruses too. And when a virus lands on the surface of a bacterial cell, what it does is to inject its DNA into the cell. And once that DNA gets into the cell, it has the information to make new viruses. And so the bacterium at that point has very little time to fight the virus before the cell dies. And so over evolutionary time, bacterial cells have come up with lots of clever ways to fight these viruses. And scientists have been, uncovering these over the years. And then about 10 years ago, a colleague of mine at the University of California, Berkeley, Jillian Banfield, called me in my office one day and told me about a really interesting finding that she and a few of her colleagues had made by sequencing the DNA of many different kinds of bacteria. And that just means reading the actual chemical code in the DNA of those cells and looking to see what was there. And what they had found was that many bacteria have inside the bacterial DNA, so this circle here shows you the bacterial chromosome in the cell. Many of these bacteria have an interesting set of sequences in the genome that includes a series of repeated DNA sequences, shown by the black diamonds, that are sitting next door to unique sequences that come from viruses. And this was the first hint. These sequences had come to be called CRISPRs, and this was the first suggestion that these might actually be some kind of immune system operating in bacteria. And we speculated at the time that these sequences might somehow work together with CRISPR-associated or Cas genes that encoded proteins in these same bacterial genomes. But at the time, that was a hypothesis. Nobody knew if that was true, and we also had no idea how that kind of immune system might actually work. So the reason that Jill Banfield called me to tell me about this was that she knew that my laboratory had a long-standing interest in another kind of molecule called RNA that is a chemical relative of DNA. And I want to very briefly tell you about that. So this is what's known as sort of the fundamental basis for molecular biology, which is that in cells we know that DNA, the double helix over here, encodes the information needed for the cell to grow and divide and develop. But the way that information is used is by first making a copy of a piece of this DNA sequence in the form of molecules of RNA. So these have the same types of letters that can form a sequence in this molecule right here. And that really is a code. It's a coded message that allows the cell to read this code and make it and turn it into a protein over here. So this is kind of the fundamental flow of genetic information in cells. My lab has always been interested in examples where the code stops right here. In other words, the cell makes a little copy of a DNA sequence in the form of RNA, but instead of then using that to make a protein, it actually uses the RNA itself to do something interesting. And we've always been fascinated by that process in evolution and how that has maybe led to the evolution of different kinds of cells. And so our idea with the CRISPR sequences was that maybe cells, maybe bacterial cells, were actually using these CRISPR sequences as an immune system that was functioning by making RNA copies of the viral DNA bits that were stored in the CRISPRs of these bacteria. That was our hypothesis, and that's why Jill asked me if we would like to do some research on this possibility. So we started working on this, and eventually it led to a wonderful collaboration, international collaboration with Emmanuel Charpentier's lab, and we figured out that one of the key proteins that works the way that this system really operates in bacteria is by a protein that's called Cas9. And in this picture, you're looking at a 3D printed model. So we take a 3D printer, and we actually can print a molecular model of what this would look like if we could see it with our eyes. Of course, this is a very tiny, tiny molecule, so we could never actually see it. But it's helpful to make a model like this so that we can think about how it works. And this is based on a real molecular structure of Cas9. And so in this model, what you can see is the Cas9 protein in white holding on to a piece of RNA that comes from the CRISPR locus. So it's a little copy of a viral DNA sequence that was stored inside that CRISPR part of the genome of the bacterium. And this protein RNA complex searches through the cell to find a place in DNA that has a sequence of letters matching the letters in the RNA. When that match occurs, the protein grabs onto the DNA, like you can see in this model. It actually opens up that DNA double helix and allows the RNA to interact with the DNA. So it makes a very tight association with the DNA at a particular place. And once that occurs, there are two chemical cleavers in the protein that cut the DNA. So for bacteria, this is a great way to find and cut bacterial, viral DNA that has gotten into the bacterium and basically lead to the destruction of those viral DNAs. But we realized when we saw how this protein was working that we could use it for a very different purpose inside of cells like our own cells. And I want to just briefly explain how this works. And the idea is that in human cells or plant cells and many other kinds of cells, when a break occurs in the DNA in the cell like this, cells have ways to repair that break by making a very precise change to the DNA sequence at the site where that double-stranded break occurred. And so we realized we could actually use this Cas9 protein to create a break in the DNA, and we could, and very importantly, we could program the protein with an RNA that would take it to a site where we wanted to make a change. And that's the key here. So the same protein can be programmed over and over with different RNA sequences that allow it to go to essentially any DNA site, DNA sequence in the cell to allow it to be changed. I'm going to show you a very short movie here that illustrates how we imagine this might actually work inside of a cell. So we're looking, oops, sorry about that. So we're zooming into a cell, and you can see that this is a cell like, let's say, a human cell that has the DNA inside of the nucleus. So we're going to zoom into the nucleus, and there the DNA is wrapped around proteins called histones. So you can see it all packaged up like this. And so the Cas9 protein with its RNA guide searches through the entire DNA of this nucleus to find a single place where there's a sequence of letters in the DNA that matches the letters in the RNA. And when that occurs, it grabs onto the double helix, cuts the DNA, and then hands this broken end off to other proteins in the cell that can repair the break, sometimes by actually pasting in a new sequence of DNA. And so you can imagine if I just cut the DNA at a place where there was a mutation, I could actually trigger the cell to correct that mutation by this kind of process. So scientists are now all over the world have adopted this exciting technology to make precise changes in the DNA of many different kinds of organisms. So for example, it's now possible to make mice that have precise changes to their DNA that makes them better models of human disease, so we can study the effect of drugs that might be curing them of disease. Also in other types of animal models like monkeys. We can also do this in cells, and if we do it in cells that are called stem cells, then these are cells that have the ability to develop into different kinds of tissues. So the hope is that one day we might be able to make changes to stem cells from patients that will allow them to be implanted back into someone that has a genetic disorder and allow them to have a very powerful treatment or even cure. And this is technology that works not only in humans, but also in many other kinds of organisms, including plants. And I wanted to just show you that already scientists are making modifications to various kinds of crops and foods that we consume, including a recently announced CRISPR mushroom that has a mutation in a gene that causes the mushroom to turn brown when you cut it. So this is now a mushroom that when you cut it, it doesn't turn brown. And in the United States, this is this type of editing using CRISPR that doesn't introduce any foreign DNA into the resulting plant or crop, like this mushroom, is not something that our government is regulating. So there are a lot more of these types of products in the pipeline. It's going to be a very powerful impact on agriculture in the future. And finally, I just want to mention that one of the uses that I've been very involved in discussing publicly is the potential to use CRISPR technology in sperm, eggs, or embryos. These are cells or very early embryos that could have changes made to their DNA that would then become heritable. That means those changes would become a part of that resulting organism, like a whole person. And not only that, those changes could be passed on to that person's children and their children. So it really means that we now have the ability to change human evolution because we can control our own DNA. It's a very powerful thought to think about. It's also a little bit scary because we can imagine that there might be lots of applications of this that might raise ethical questions. And so I've been really engaged in helping to discuss this, educate people about it, and invite a global discussion around appropriate and safe and ethical use of CRISPR technology, especially in the human germline. This was actually a cover picture taken for The Economist magazine that came out last year. The title of the article was Editing Humanity. And this is really just showing you some of the things that could happen in the future as we understand more about the human genome and have the ability to make changes that not only correct disease mutations, but also introduce properties that we might consider desirable. And the big question for all of us, and especially for all of you, because it's really your future, is should we go there? And how should we ensure that a powerful technology like this is employed in ways that we all feel comfortable using? So I'm going to close there. I just want to jump to this slide here. Huge thanks to a very wonderful group of people in my lab. This is a picture taken at a recent baseball game in San Francisco. We went to see the San Francisco Giants. And I have people in the lab from really all over the world that have come together to work and do science together. And like I said at the beginning, I think for me that's one of the most wonderful things about science, is being part of a global community of people and collaborating and working together to really understand the fundamentals of our world and then figure out how to harness that knowledge for the betterment of humanity. Thank you very much. I'll be happy to answer any questions.
Thank you so much for the sharing presentation. Please return back to the seat. Thank you.
Okay.
Move on, we will have the Q&A section. We will first invite two delegate students from our school to have the conversation with Dr. Doudna. Let's welcome Dr. Doudna, Principal Yang, and the student Jing Shen and Peng Rui to come to the stage.
在座所有的同学们，很抱歉啊，因为我们学校是地主队嘛，哈，所以前面呢，我们会有2个我们学校的同学们，他们先看过了 slide 之后，会先想到一些问题。等一下，他们2位先发问，他们发问之后，接下来下一个段落，我们会开放给所有的在场同学们发问，这样可以吗？可以，谢谢你们哈，谢谢 ，OK。
So first of all, I would love to thank you first for the marvelous and inspiring speech, Dr. Doudna. And that makes me wonder how CRISPR accomplished such incredible precision, efficiency, and success rate.
So that's a great question, and it really, I think, goes back to where this technology comes from. It's a bacterial immune system, meaning that it's sort of a battle between a bacterial cell and a virus that would like to destroy it. And so this CRISPR-Cas protein and its RNA guide have evolved to be very efficient. They have to be very good at finding viral DNA and cutting it up before the cell gets killed off. So there's a natural selection for this to be a really efficient, fast system for finding and cutting DNA.
And another thing is that it is programmable. But how does that really work? How do scientists program it?
Right, so that's a great question. So scientists can program it because we understand how the RNA is made naturally in bacteria, and then how we can make it in the laboratory and make it with any sequence of letters in the RNA that match a sequence of letters in DNA where we would like to introduce a change.
First of all, Dr. Doudna, I would really like to appreciate your inspiring speech. It really benefited me a lot. And there is one thing I have been wondering about. It seems like this is such a powerful and influential technology that can be widely applied on monkeys and mice and even human beings. But if we apply these CRISPR systems to humans, will there be any limitations?
That's a great question. And the answer is that today, the technology is still developing. So if you look at what kinds of experiments are being done worldwide, there's only a few countries that have allowed scientists to do experiments in human embryos right now. So it's still being figured out how efficiently it will work there, and whether it will really be suitable for making changes in embryos. But if we assume that will happen, I believe that it will, then that really opens up to, I think, a fundamental question that's not about the ability of the technology, but it's about our own ability as people to make decisions about using something that could be used in ways that would be ethically, I think, challenging.
So you also mentioned in your speech that the CRISPR system can be used to cure diseases such as cancer. So if we apply that to the human body, how are we going to be able to target the infected cells and not just editing all the genomes in every single human cell?
Yeah, so you're asking a very, very important and really good question, because I think that's a key bottleneck right now. It's a key challenge that all of us as scientists are facing, is we have this great technology that we know works well for editing DNA in the laboratory and in cells that are growing in the laboratory. But what you just said is the key question. How do you then deliver it into tissues where you'd like to make changes that would be beneficial to a patient? There are many people, labs in academic organizations, also companies that are working on that challenge. And there's a few ideas. I think that one of the early uses of the CRISPR technology will likely occur in blood cells, because those are cells that you can, in principle, remove from a person. And if there are some stem cells in there, and that means cells that have the ability to regenerate, then if those were edited, then they could be put back into the person, and they would continue growing with the modification in the DNA. So I think that's a place where you kind of get around the delivery problem. But if you really wanted to use it to, as you said, sort of try to treat cancer, I think what you'd want to be able to do is to do editing in immune cells, so that you would basically teach immune cells how to track down and kill cancer in a patient. That's going to be harder, because we'll have to figure out how to get these editing molecules into the immune cells in the first place.
So the last technical questions. Besides CRISPR-Cas9, what are other topics in the field of biotechnology or biochemistry that you've been interested in or you consider promising in the coming future?
Well, I think one of the things that I've been fascinated about is that if you look at how this CRISPR technology came about, and frankly, a lot of the fundamental technologies that we're all using in molecular biology, they've mostly come from bacteria. And so I think that's telling us something very interesting and fundamental about nature, which is that bacteria have been around, frankly, a lot longer than we have, and they live in a wide range of environments, and they have to deal with all sorts of viruses. And so that means that there are a large number of solutions to the problem of viral infection that have evolved in these simple, single-celled organisms. So I'm very excited about the potential to continue mining what I call the bacterial universe, figuring out all the, we've only been able to study in the lab a tiny, tiny fraction, way less than 1% of all of the bacteria that exist on our planet. So I think there's a lot of interesting research to be done by investigating different kinds of bacteria.
So now let's move backwards a little bit, back to your high school days. So what motivates you? What sparks your intellectual curiosity? And how does that zeal influence your academic and later on career path?
Well, as you heard at the very beginning, I grew up in a small town in Hawaii, and nobody in my family was a scientist. My dad was a professor at the University of Hawaii, but his specialty was English literature. So very different field. But I think I was really captivated by the island environment there, kind of not so dissimilar, frankly, from Taiwan, in the sense that there were lots of interesting evolutionary changes that had happened to organisms growing there over time. And I always wondered as I was growing up, like, why are there blind spiders in caves, and why are these really cool birds that don't fly anymore because they've been evolving in this island environment. And so that kind of was maybe my underlying inspiration. But frankly, when I was in 10th grade in high school, I had a wonderful teacher, Miss Wong. I still remember her, Miss Wong. And Miss Wong was my chemistry teacher. And she was really gifted at telling us kids that science wasn't about memorizing a bunch of facts in a textbook. That's not science. She said what science is about is it's about solving puzzles. It's about asking a question and then figuring out how to figure out the answer when no one knows what the answer is, right? Nobody knows the right answer. You have to figure that out. I found that to be super exciting. And it just really captured my attention. And I think it was really then that I thought, that's really what I want to do with my life. I want to apply that kind of chemistry and that kind of questioning nature and trying to understand, figure out the answers. I want to apply that, but I want to apply it to biology and cells.
So has it always been dreaming of becoming a scientist in the field of biochemistry, among other options?
Well, I had a few, that was my early idea, but I had a few definitely moments when I questioned that. When I got to college, I struggled. My first year in college, I was taking a general chemistry class, and frankly, really struggling with it. And I had come from a big public high school in my town in Hawaii, and we just didn't have the kind of training that a lot of my classmates in college had. So I found myself, really, really struggling and studying hard and not doing well on exams, and really asking myself, do I really have it? Do I have what it takes to do this? And I was taking a French class, and I loved my French teacher, and she was just really, really a very engaging, kind of warm person. And so I went to see her one day, and I said, I'm thinking about changing my major to French. And she said, well, what's your major now? And I said, chemistry. And she said, oh, stick with chemistry. So, I think it's sort of an anecdote that illustrates something that's happened to me multiple times, frankly, is when I've had doubts about myself. And we all do, right? You have doubts. You wonder, gosh, am I really on the right path? And then, am I really going to be able to do this? I was very grateful and fortunate that I had people in my life that would tell me, yes, you can do it. If you want to do it, you can do it. Don't let anyone tell you that you can't. And that's really the message I would love to convey to all of you. I think that's really been true in my own life, is that, I come from kind of an unlikely background, to be doing what I'm doing, and yet I'm doing it. And why is that? Well, it's just because I always wanted to do it. I really, really wanted to do it. And I just never let anyone tell me that I couldn't do it. I kept, in my own mind, I kept saying, I know if I work hard and I try to do this, I'll be able to figure it out.
And so how do your academic backgrounds contribute to your achievements today?
Well, again, I think, like I said, I went to not a fancy high school at all. I was a, public high school in my town, and, it was a mixed bag. I had some really great teachers. I had some not so great teachers. Most of my classmates were not going on to college. I think very few of them did. Most of them were going to stay right in my town of Hilo, Hawaii, and, do whatever. They weren't sort of going on, in sort of fancy professional careers or anything. But again, I feel like it's really just about, deciding what it is that you're passionate about doing, and then going after it, just really embracing it. And when I look at, students that I've had in my lab over the years, those are the ones that, I've had a lot of incredibly bright, really, really talented people. I would say, in general, the ones that do the best, though, are those that are really, really dedicated. They're really passionate about what they want to do. And they just stick with it. They're really perseverant. I think there's something to be said for just, really deciding, I love this, I'm going to figure it out, and I'm going to do what it takes to make it work.
So, as a female scientist, have you faced any obstacles during your research process, and how did you overcome them?
Well, I think for women in science, or girls, girls and women in science, I think that, what I've seen is that, there have occasionally been people that have said, well, girls can't really do science. I mean, I guess the example I'm thinking of as I'm saying this is I had a high school guidance counselor in Hawaii who actually, I went to see him. We were required to go to our guidance counselor, and I went to see him, and when he found out that I said, I'm interested in chemistry and biochemistry, he said, well, that's not a good subject for girls, And what he didn't know about me was that I'm a very stubborn person. And if someone tells me I can't do something, that makes me say, I'm going to do it. I'll just show him. So, but I think, that there have been occasionally things like that have happened to me throughout my career. And, my approach is to just say, well, that's fine if someone wants to think that. But, if I think in my mind or in my heart that I want to do this, I'm going to figure it out. And the other thing is that I've had a really, really great set of friends and colleagues that have been supportive. And I want to especially give a shout out to my husband. So I'm married to a scientist, and he's also a professor at the University of California, Berkeley. And he's always been incredibly, incredibly supportive of me, and helped me through thick and thin and problems that come up. And this is a trip now that I'm going to be on the road 2 weeks, and we have a 13-year-old son at home, and so my husband is home with our son and helping him with his homework at night every night, and just all the being a great dad while I'm off having fun, right? And so, but I think that's something that I actually tell both my male and female students often, is that I think one of the things that's really important to success in science or whatever you're doing is pick the right life partner. Very important.
So since a lot of students who attend this lecture today are females, do I have this honor to invite you to give us some encouragement?
Of course. I think I already have, but basically I just would say to especially the girls here that I encourage you to figure out what you're passionate about doing. If it's science, that's great, and you can definitely do it. If you want to pursue a career in science, it's just a matter of putting your mind to it, and don't let anybody tell you that you can't do it, because you can. Just look at me. I didn't think I'd ever be sitting here. Believe me.
我刚刚听到以后觉得非常的有趣啊，因为他们问什么问题我不知道，我刚刚从这个现场这样听到， 他们问了2方面的问题，一个是关于他的研究方面的问题，第二个部分是关于他的生涯发展，还有就是一位女性科学家啊，她碰到的挑战。在座有这么多的女性同学哈，其实对男同学一样，好好找一个人生伴侣是非常重要的，他要好好支持你哈啊，这是第一点。第二点就是这位教授他在大学时代，他也曾经考虑过不要学化学了，因为大一普化不好学，是不是干脆去学法文算了，对不对？好，我看到我们的化学老师点头，对，不好学哈。所以有一些时候，他有提到他的那个固执、坚持下去哈，以及有一股傻劲。那第三个是这2个部分，我相信都会给我们很好的一些鼓励哈。那关于他的研究的部分，是我今天很抱歉，因为我很忙，我今天我才特别听到，才去研究。我之前就看到这个 CRISPR-Cas9， 我一直在想这是什么东西，这是什么东西啊？因为我自己是学辅导，不是学这个领域的。我听到它是从细胞里面得到的一些灵感，怎么样去找到这个序位，影响到我们的 DNA 要去做这个传递的时候。我觉得哇，这个发展的空间真是无限，将来说不定各位就可以真的制造出那么样一个优秀的小孩，高 IQ， 视力2.0，不会秃头哈，这个未来都靠大家，还有治疗癌症啊，所以好好的提问，希望让我们今天下午呢，在这里真的是非常愉快的。我相信我们在座有很多同学，你们都会有一些很棒的问题。OK， 那我们就会开放让你们提问。OK， 好，开始了。
Good afternoon, Professor. And I have some fellow question. First is, I'm wondering, like in U.S., some legislation have a little bit constraint about like scientific progress. Like you can't use some embryo or you can't cut some genes. And I'm wondering, like, whether the legislation in the U.S. have limit your research project.
So in the United States, the rules right now are that you cannot use government, federal government money to pay for human embryo research. But if you have another source of funding, like let's say state money or money that's donated by a private person or a foundation, then you can. So it's kind of an interesting, situation where you can do it, but you have to get funding that is not coming from the federal government. But that being said, my own work is very much focused on molecules. So for the most part, in my own research, we're working on things like, understanding how these molecules work, and then finding other ones and testing their behavior in cells, rather than doing anything with human embryos. So it certainly hasn't affected my own research, but I do think that going forward, there's a need to ask whether the regulations in the United States are, is that the way we want it to be? Because it's certainly different in other countries, as you probably know.
Sorry, another question. Like, because you have mentioned in your lecture, like some stem cells, if we can really create an ideal baby, like 100%, they are smart, they are brilliant, they are pretty. So is there any like further limitation about this research? If we can prohibit some overuse of this fantastic science?
So I think one thing that's really important to appreciate is that even though we have a technology now for making precise changes to the DNA of human embryos, we don't know, there's a lot of things that we don't know yet about it. And one of the biggest ones is that for the most part, we don't know what genetic changes would be necessary to do all of the things that you just mentioned. So, that knowledge will come over time, but today, we don't have it. And I really feel like to do the kind of thing that you just mentioned is probably still, a couple of decades away, really. But I guess my point in discussing it really is just to point out that it's coming, that time is coming, and it's not a fantasy anymore, right? It's not a science fiction. It will happen, and it will happen for sure in your lifetimes. And so it's really important to be already thinking about what are the right ways to use this, and how do we ensure that it's used ethically.
Okay, last one question. Sorry. I'm wondering, like, in the most important core of science researching, do you think that curiosity is the most important, or knowledge?
I think curiosity is really important, honestly. I mean, you have to have enough knowledge that you can be curious, and curious in a way that allows you to actually answer those questions that you're raising, right? But I think that having an open mind and really just thinking deeply about nature is a really important quality for scientists. And frankly, I see that in the youngest kids. You know, I think very young kids they're natural scientists. They're curious, and they're open-minded, and they're asking interesting questions. And then they just need to gain enough knowledge that they can actually act on those questions in the future.
Thank you so much.
好，接下来我们外校同学优先好了，好不好？我看到这边有一位建中同学举手发问。
Good afternoon, Professor. I have a question about the CRISPR technology. I'd like to ask if there's a way, if I want to CRISPR my cell, whether the protein and DNA can enter the nucleus, or it has to be, or the genome has to be changed when the cell is replicated.
Okay, so this sort of gets back to a question that was asked earlier about how do we introduce these editing molecules into cells or tissues. And right now, that's a big challenge, is how do we actually do that? So, you know, there are a few strategies that people are using, like using a virus to deliver it, or we can use various ways of enclosing it in a little capsule and then delivering it that way. So those are a couple of the approaches that are being used. And then with regard to your question about getting into the nucleus, it turns out that you can put a little signal, a little chemical tag onto the protein that allows it to be taken up into the nucleus. So it's almost like we would call it in the US a zip code. You know, it's a little code that tells the cell this is something that should go into the nucleus, and then it does.
Very thank you.
First of all, thank you for your inspiring speech, but I'm a little bit confused that is there any differences from these CRISPR technology from the gene transfer technology? Is there any differences from these two things, or is just the promotion of gene transfer?
Well, I guess I'm not quite sure what you mean by gene transfer, but I think the thing that's important to understand is that, you know, scientists have been coming up with better, you know, different ways to manipulate DNA in cells for a long time, right? This isn't the first technology to do that. What's special, though, now is that you can do it very precisely. So we're not relying on, say, a virus to randomly insert a sequence somewhere in the human genome. We're actually saying, I want to make this change to this letter in this part of the DNA code, and that's the only change I want to make, and we now have a technology that allows us to do that.
Okay, thank you.
谢谢。这边的同学，好。
My question is, this February, the UK gave official approval to the scientists that they can use CRISPR-Cas9 to edit human gene, and that may cause a huge influence of human being. And do you support the experiment? And what do you think about this issue? Thank you.
So so there actually are now 4 countries that are allowing scientists to use the CRISPR technology in human embryos. But it's important to explain exactly what is being allowed. And basically, in those countries, what's being allowed is for scientists to use the CRISPR technology in human embryos up until 14 days. Okay, after 14 days of development, those embryos must be discarded. It's not legal anywhere right now that I'm aware of to implant those embryos into a woman with the goal of creating a person from those edited embryos. So I think the important purpose of these ongoing experiments in human embryos, and frankly, I do support this because I think it's an important research project, is to understand enough about early human development. It turns out we don't really know that much, amazingly, about our own, very early development and how a gene editing tool like this would work in a series of dividing cells in an embryo. So I think to me, that's really important research that needs to be done to allow us to answer the question, do we ever want to use this in embryos for clinical use, where we would implant the embryo? I think we can't answer that question unless we have enough data to know how it would work.
Thanks a lot.
好，来，这位同学，对。
I'm from Chenggong Senior High School. And I really appreciate your making time in your schedules to attend today. And thank you for giving me this opportunity to ask questions in this special occasion. Frankly, I know that it's impossible to create a children that has a IQ 180, or Stephen Curry's basketball skills, or Taylor Swift's voice, and study in TFG in the same time. But I have to ask that, do you think that your discussion may serve as a blueprint for other fields? Thank you.
Thank you for that question. I think that maybe I think what you're asking me is, do I think that having an open discussion about the use of a technology and thinking about the ethical implications of that, will that invite other scientists and frankly all of us to engage in those discussions around other technologies as well? And I certainly hope that that's true, right? I think what we've seen, what I've seen in my lifetime. So I'm 52, okay, and what I've seen so far in my lifetime is that there's been, I think, a decrease in people trusting scientists, and trusting science. Because when I was a kid growing up, we thought scientists were amazing. I mean, I did. I thought science that seemed incredible. I wanted to be a scientist because I thought, that would be a way to really contribute to the world in a positive way. And I think what's happened now is that, like when I look over the last 10 years, especially in the U.S., there's a lot of people that they don't want to vaccinate their children against disease. They don't believe in climate change. They reject genetically modified organisms because they don't trust science and scientists. So, and there's many other examples like that, right? So I think it's been a real problem. And when I ask myself why has that happened, I think part of it is, frankly, the scientists' fault. I think we're kind of at fault because I think for many scientists, and I'm this way too, we'd rather do the next experiment in our lab because it's fun rather than, get out and really discuss topics that might be kind of uncomfortable, right? It doesn't get much more uncomfortable than talking about human embryo editing, especially if you're like me and you don't know much about it, right? But I think it's really, really important. So I kind of made a decision a few years ago when I could see where this was all going with the CRISPR technology that I needed to not do that, and I needed to really be taking a public stand and encouraging an open discussion. And I do hope that will encourage others, scientists, and frankly, the rest of us to do the same.
Okay, thank you from the bottom of my heart for giving this chance to speak to you today.
Thank you.
Thank you.
我特别有感觉，因为这个题目非常有趣啊。因为刚刚帮我翻译一下，就是他刚刚提到以及教授的这个回答啊，因为教授的回答里面提到，他小的时候大家对科学家比较信赖的。其实我自己也活了跟他差不多接近的年纪，我觉得现在各个领域大家都不太信赖，所以不管是在哪一个领域啊，各位可能将来成为科学家、政治家、老师，我觉得永远都不能够忘记一件事情，就是我们要想到他人的这个利益啊。如果我们能够对人、对物、对自然有更多的尊重，那么我们彼此之间的信赖才会足够，对不对啊？ 那科学也才能够对我们的人类有更大的贡献。OK， 后面我再看看，对不起，我先看一下有没有其他学校的同学发问。好，我们先给一个机会给师大附中的同学，好吧？因为成功发问了，建中发问了，中山准备哈。
Good afternoon, professors. Thank you for your amazing speech. And my question is that could we just deliver the cell into an adult human directly and then we can cure some disease that caused by genetic disorder? Thank you.
Yeah, it's a great question, and it's definitely one that many scientists are working on right now, which is, can you take stem cells that would be able to regenerate a tissue and make genetic changes that would correct a disease-causing mutation and then put those back into the patient. And I think the example that you may have heard of this already because it's talked about a lot is the idea of being able to cure diseases of the blood, like sickle cell disease, which causes very painful changes to red blood cells that is a disease that's caused by a genetic change that we know about, and up until now we haven't really had a way to fix it. And now, in principle, what you could do is take blood stem cells from someone that has that mutation, correct the mutation, and then put the cells back into their bloodstream. So, I think that type of use of the CRISPR technology is likely to be the first type of clinical application that we'll see. But, there's many other opportunities, especially as we become better at being able to deliver these molecules to other kinds of tissues too.
Thank you.
好，我的眼光一直看到我的左前方，有没有同学要发问？有，好，果然看到了。来，我们给中山女高同学一个机会，好吗？啊，谢谢。
Good afternoon, Professor. So my question is that where do you see the CRISPR technology in the future?
I think that it's going to have a really big impact in three kind of big areas, right? One is human health, as we've been discussing, for making, I think initially at least, for making changes in, not in embryos, but in adults, in people that have a genetic disease and figuring out how to cure them of that disease. I think that's one very important area where it'll have a big impact in the future. The second one is agriculture. It's going to be really, really important in plants. And the thing to think about there is that, we are facing global climate change. We're also, we're living in a globalized world where plant pathogens, meaning, fungi and things like that can affect and destroy plants, get around the world really fast now, right? And a lot of plants, because they've been cultivated for a long period of time, they don't have natural genetic defenses against those kinds of organisms. So I think being able to use genome editing to help those plants protect themselves from pathogens, that's going to be really, really important in various parts of the world for, frankly, for the food supply. And then the third area is in what I would call synthetic biology. And that means being able to make genetic changes in organisms that are helpful to us because they make useful molecules. They make biofuels, or they make chemicals that would otherwise have to be produced in a very toxic way. So I think those are really the three really big areas where this kind of technology is going to be important as an application. And I just want to add that, the other thing to think about, especially for those of you that might be thinking about going into science in the future, is that for scientists who want to understand fundamental biology, just about, how does the world work, right? How do organisms function? This is an incredible research tool, right? So because it basically makes every organism now a genetically tractable system. So you're not wedded to a few model organisms that traditionally, geneticists have had to use to study life in the lab. We now can, in principle, we can study any type of organism that we're interested in because we have a tool that lets us make very precise changes to the DNA and ask what happens when I make this change.
Thank you so much.
好，我们还有时间，显然没有人阻止我再点同学们发问哈。好 ，OK， 刚刚后面有同学啊，我们要让后面的同学有机会，好吗？ 麦克风能不能够传递过来一下，让我们后面的同学有机会。
Good afternoon, Professor. And my question is that in the beginning of your speech, you mentioned that you were excited about exploring the potential of like unknown bacterial genomes. Like you can maybe find the cures for uncurable diseases inside their hidden DNAs. But when you play around with genes, you're actually changing the ecosystem of a lot of creatures. You're actually changing the whole ecosystem of the universe. So my question is, isn't that sort of controversial to maintaining biodiversity?
Well, you know, if you think about it, evolution has been doing that for a really long time, for a lot longer than we've been around, right? So, you know, the way that natural selection works is that, you know, there are random mutations that are introduced all the time. They happen in our DNA, they're happening in plants, you know, other animals, basically every type of organism that are being introduced by, you know, radiation that's coming in, chemical, you know, interaction with chemicals and other things. And so, you know, that's kind of the way natural selection works, is that it operates on this pool of mutations, and if now and then a mutation, a random change occurs that happens to be beneficial, then that trait, that gene, tends to be selected for over time. So I would say that, you know, that's something that's been going on all the time. That's kind of the basis for life. And what this gene editing technology does is it gives us a way to do that in a controlled way, right? And so that it just gives us a way to understand how evolution is working, because now we're not waiting for a random mutation to occur. We can actually make a change and then say, what does that do to that organism?
Thank you.
真是感谢大家这么踊跃的提问哈。好，那我们现在中间有一位同学哈，谢谢。
Good afternoon, Professor. And I really think CRISPR is such an incredible research. And my question is that, like as you mentioned, maybe we can make a perfect baby in the future by the CRISPR technology. So a real human be the subject of the experience, or if like people experience is not allowed, if you use like a mice for an alternative, how can you like ensure that the DNA change will have the same effect on a mouse than as on a human being? And if like human experience is allowed, how to deal with the like ethical problem? Thank you.
Yeah, it's a really important question. So I think you make a very good point, and that is that, you know, we can do experiments on animals, but in the end, we don't really know for sure that data that we get from animals will apply exactly in humans. And as you may know, that's been really the experience of many drug companies, right? When you try to use mice, let's say, as an animal model to test drugs in, you know, sometimes you have drugs that work great in mice, but then when you start testing them in human patients, they're toxic, or they don't work at all, or they have other problems. And so, you know, you're absolutely right that I don't think you can, you know, be sure that if you see a result in an animal system that it will translate exactly into humans. So that does raise a really, really important question, which is, you know, if you ever wanted to use this in humans, especially in embryos, where the change becomes permanent and inheritable in future generations, how could you ever really do controls? How could you do studies that would ensure safety? Because you can't really do exactly what you might want to do to get the data you need. And I don't know how to answer that question. You know, it's a tough one. I think that that's where these real ethical challenges come into play. And you know, I have to tell you that I get emails daily, really every single day, from people around the world that are facing genetic disease in their families, and they're really desperate for anything that can, you know, help them, right? And they almost uniformly, people in that situation, they really, really, really want this technology, and they want it right away, you know, because they're really, you know, they're really desperate. And my heart really goes out to them, because, you know, I totally understand that feeling of, you know, especially if you have a child that's, you know, suffering, right? You want something to help them. So I think that's going to be on the one side, and on the other side is going to be the issue of safety, you know, and ethics, and it's just going to be something that we're going to have to grapple with going forward. I don't think there's an easy answer to it. I think it has to start, though, with as we said in answer to an earlier question, I think it does have to start with appropriately regulated experiments that are done in early human embryos so we can understand how this technology works, at least in that setting. That's at least a place to start, I would say.
Thank you very much.
好，后面这边还有一位同学，我看到了，前面有一位那个连坐在地板上都要发问，等一下一定让你有机会，哈哈，真是太感人了。
Good afternoon, Professor. Even if your husband can take care of your son like now, have you ever struggled between your family and research? How did you balance between them? Thank you.
All the time. It's a constant struggle, and everybody I know that is, married or, just in a family situation of, however their family is structured, they face it too, because, you're juggling your work responsibilities with your responsibilities as a parent and as a partner, and there's no sort of, at least in my experience, simple formula way to answer that. It's for me and for my family, it's just sort of it's an ongoing negotiation. It's an ongoing struggle where you just try to if you feel like you're working too much, you try to take a little more time with the family, and go the other way too. And I think what I find is that in my work, my work really tends to go in cycles. There are times when we're working really, really intensely on a project. I mean, the Cas9 project, frankly, was like that, right? So, that project kind of started, and right away it looked really exciting. And so that, just really interesting data were coming in. And it was almost like a crescendo, right? Where you're just, you're working, and, and then, writing a manuscript about the data. And, Emmanuel was in Sweden at the time, and we were, emailing back and forth at every hour of the day and night. And, but then once that, that paper was published, then there's a little lull, right? And, and so then I was able to spend more time with my son at that point. And, so it's always kind of just a balance, and trying to not let your life get, pulled too far in one direction. But like I said, it's just, I think it is honestly more, it's harder for women, just because I do think that, as moms, if you have children, when you're a mom, there is a stronger kind of pull with your child somehow than what I've seen for a lot of men. Not always, but I think a lot of times that's true. And it just means that you have to work harder to try to find that balance with your work.
Thank you very much.
不过我要说一下哈，因为刚刚教授说到，当妈妈很不容易。其实我昨天晚上看到一幕，我很感动。昨天各位知道，现在进入秋天了，天气转凉。我昨天看到有一位爸爸在某一个国中旁边啊，离学校有一段距离，不是就特别指我们学校旁边国中。他晚上帮孩子递上外套，然后骑摩托车，两个人一起回家。那一幕让我觉得好温暖啊！因为在这么忙碌的台北的工作环境之下，他晚上来接他的小孩，那种感觉让人家觉得这个才是生活，因为生活离不开人性啊，所以两边之间保持平衡，永远是我们要去做的事情。OK， 好，那我们接下来要给那个在地板上还要提问的同学，谢谢你啊，为难你了哈。来，麦克风现在在哪里呢？谢谢。
Good afternoon, Professor, and thank you for inspiring speech. And my question is, do you think that the unique differences between every person will be affected as the technology is being used as creating a perfect child or a perfect person?
Well, I think it will be a long time before that might happen, right? Because it takes a long time for a baby to develop, and as we've been discussing, the CRISPR technology is not today ready for that kind of use, and we don't know enough about the changes that you would put into the DNA of a person to make the quote perfect baby. And I would argue we probably all would have a different definition of the perfect baby. Maybe we would. But anyway, I think that so I'm less worried about that. But I do think that
that it's an important issue to be thinking about, because what I suspect will happen, honestly, is that I think as this technology continues to develop, and by the way, as you can probably imagine, this field is just moving at lightning speed, right? It's moving very, very, very fast. And this is a technology that's widely available, it's inexpensive to use it, and it's not that hard to use it. So it just means that many, many, hundreds, probably thousands of labs around the world are already using it, some of them for the purpose, for thinking about how to use it in human embryos. And so I think that means that, over time, the capabilities will increase and get to a point where one could imagine using it to create a genetically modified person, basically. And so, this is why I think that it is really key to be grappling with these issues now, even though I frankly don't think that, I think we're still probably a couple of decades away from, that being kind of a reality of being able to make a lot of changes in a person, and have those changes create the kind of features that you would want.
Thank you so much.
好，我们还有一些时间，还可以继续来听听看同学们的发问。好，我们给男同学机会，要让他们优先。好，来这边。
Good afternoon, Professor. I'm honored to listen to your speech. I'd like to ask, I found a news which says that when CRISPR system goes to T cell, one has been infected by HIV. Some of the virus had hid from the capture of CRISPR. Do you have any idea of this solving this problem? Thank you.
Well, I don't know about that particular example, but what I can tell you is that I think there is a really interesting way that the CRISPR technology and similar types of genome editing in general can be used to treat HIV. And it's actually a treatment that's already in clinical trials for humans. And the way it works is that instead of trying to target the viral DNA that's already infected someone's T cells, the strategy is to make changes to the DNA of T cells before they get infected to make them uninfectable by HIV. Okay, and it turns out there's a natural variation, genetic variation, that occurs in a very small percentage of humans that makes their T cells uninfectable by HIV. And so once that was figured out, then scientists realized they could actually use genome editing to introduce that change into people that don't naturally have it, to make their cells resistant to HIV infection. And that's actually an approach that's I think it's already in phase 2B clinical trials. So it's really, progressed fairly far. And I think that could become a treatment that will be approved at some point in the future.
Thank you very much.
那我们麦克风就交给后面这位同学，好吗？好，然后等一下我们这边有同学发问，我刚刚有看到，哈，谢谢。
We're delighted to hear your result of your research, and I think many of the people here may inspired by your story or result. Can you give us and your children a suggestion of how to face the future, how to broaden our sight?
I think all of you are sort of coming to the point in your lives when you have enough knowledge that you can really start to have a real impact on the world. And your challenge is to figure out what your passion is, what you're good at doing, what you enjoy doing, and then bring those together in a way that is going to enable you to tackle challenges that are facing human societies. And there are lots of challenges out there, right? There's a lot of really good challenges to go after. And I think for all of you, and I say this to my son too. He's a bit younger than probably most of you, but but not that much, right? He's 13, and he's just starting to think about like what might he want to do in the future. And I always tell him, think about the big problems out there, and think about how you can contribute to solving them, because we need all of you, right? The world has a lot of problems, and we need bright, young, eager minds to go after them.
and to think creatively about how to tackle them. And I know you can do it. You just have to decide what you're passionate about doing and then go after it.
Thanks.
这个领域它未来的发展性非常的大，所以可以容纳非常多的同学们，你们将来在其中不同领域其中一个小的区块去发展，没有任何的问题。重点是找到你们的真正的兴趣所在哈。好，我刚刚有看到有同学在后面刚刚有举手的，好 ，OK， 那我们先请后面这位同学，然后前面等一下好吗？好 ，OK， 先让后面那位同学先有机会发问哈，谢谢。
Good afternoon, Professor. And to be honest, I am not really interested in science. I have more interest about, Chinese or social studies. But my parents, they are trying to make me to choose the way to study science in the future, or like study medical science, something like that. I need your advice. How can I convince them to let me follow my heart? Yeah, about that.
Well, your story is reminding me of someone I knew in college. So I knew this guy in college who was maybe in a similar circumstance. He was very smart, but his parents were very ambitious for him to be a doctor. They wanted him to be a medical doctor. And so he was taking, all the science classes and sort of pre-med classes, but he was really, really unhappy, and I could see it, right? And he was a friend of mine, and, he told me basically told me the same thing that you just said. He said, my parents are just, they want me to be a doctor, and so I'm taking all these classes to kind of please them, but it's not my thing, And I said, well, what is your thing? And he said, I love history. He says, I love history, and when I take a history class, I'm just so into it, and I just can't get enough. I want to read more, and I want to write more, and so he just, eventually he was able to express that to his parents and just show them that, he was really, really passionate. He just wasn't passionate about science. He was just really, really into history. And he ended up becoming a history professor, and he loves it, right? So that was totally the right thing for him. So I don't know in your particular circumstance, but I think that, it is important to recognize what you enjoy doing and what you're good at doing and what you like to do, because in my experience, it's really hard. Like I've had students in my lab occasionally who think they want to do science for whatever reason, but when they get in the lab, they realize, it's not their thing, right? And I can see it, they can see it. And the best thing one can do in that circumstance is just to own it, right? Just say, yep, that's who I am, and that's who I'm not, and I've got to be who I am and be true to myself. So I don't know if that's helpful, but I definitely think it's a really important thing to try to do.
Thank you.
亲爱的同学，我有一个小小的建议啊，你把今天的 video tape 拿给你的家人看 ，OK？ 你告诉他们，你在这里问了一个这样的一个问题，也得到了很好的答案。爸爸妈妈，给我一个机会，给我一点空间吧，我会证明给你们看，我做的会很好的 ，OK？ 好，我们还有最后一个机会，因为刚刚他们讲那个时间调整，必须要等一下要结束，我们还会有一张大合照 ，OK？ 哈，刚看前面有同学举手了，对不对？那个，对不起 ，OK？ 那两个好了哈，两位同学，这个前面同学，然后接着就是好不好 ？OK？ 两位同学。对不起，左边真的来不及，1分钟哦 ，so terrible.
First, I need to apologize for standing while raising my hand. And my question is that, as a scientist who studies gene and the way to transfer it, I think you understand more than other people about the risk of gene transferring. So, will you dare to eat G food? For instance, AQ advantage salmon.
Are you asking about salmon?
I mean, I just took it for example.
Yeah. So are you asking, what do I think about, say, genetically altered salmon or other kinds of foods? That's your question?
Yes. Will you eat it.
Will I eat it?
Yes, and to let your son, your families to eat.
Well, what I would say is that all of us, every single one of us here, is eating genetically modified food every single day, okay? Because human beings, no, and it's true, because human beings, we have been conducting agriculture, and frankly, it's probably been going on in China for longer than maybe many other human societies, for thousands of years, right? And what I mean by that is that humans have been selecting traits that they see that are desirable and then breeding those organisms for consumption for thousands of years. Except that in the past, up until now, we've been relying on random mutations that occurred in those organisms, right? Random changes that happened in the DNA. Because if you think about a trait that you like I like a nice red tomato that's tasty and doesn't get rotten right away. Well, those traits aren't if you look at where tomatoes came from, they didn't come like that, right? So they've been bred to be that way. And so, all of us are eating genetically modified food every single day in that sense, okay? And so what's happening now is that now we have technology that allows us to speed up that process, all right? And so now that we're, instead of waiting around for mutations to occur, we can actually introduce them. Does that, that's a powerful thing to be able to do, so it certainly has to be done thoughtfully and with appropriate regulation. But I think that it's, in the scheme of things, it's a really good thing because it's going to make it possible to, conduct agriculture in a much more rational way than has really ever been possible.
Thank you very much.
很抱歉啊，我已经站起来，因为刚刚老师已经拿出牌子来说，只剩下1分钟。显然这个回答1分钟已经超过了，所以前面的同学跟你说抱歉，还有后面的同学。今天我们在这里，我们要非常谢谢我们的这个 Doudna 博士啊，因为他回答了我们各式各样的问题，包括他的研究，包括了他的家庭生活，包括了未来。各位有什么样的一种发展的可能性？我们非常感谢大家共同在这里度过了愉快的一个下午。等一下，我们接着要拍一张大合照。同学们，等一下设法调整你的角度，一定让每一个人都被看到，好吗？好，谢谢大家，谢谢。
First, let's welcome CEO of the TangPrize Foundation to have the group photo together.